Good morning, everyone. Welcome to the annual Michigan Biology of Cardiovascular Aging Symposium. This year, as you all know, we are honoring the legacy of Dr. Daniel Goldstein, the founder of the center. This year's symposium has an outstanding lineup of speakers from diverse disciplines and expertise in research ranging from bench-side research to clinical trials. For you who had the fortunate to know Dr. Goldstein, you know how collaboration in science is an area that is near and dear to his heart. And for this year, we are looking forward to our speakers to share their cutting-edge research and fostering collaborations in the spirit of scientific inquiry that Dr. Goldstein championed throughout his career. We'll start by a short introduction from Dr. Salomon.
Thank you. Okay. Good morning. As we gather today for the annual Daniel Goldstein Michigan Biology of Aging Symposium, we want to take a moment to reflect on Daniel's lasting memory as the founder of M-BoCA. We remember Daniel as an outstanding researcher, mentor, journal editor, teacher, and friend. More specifically, today, we remember and celebrate his work to help establish the M-BoCA Institute and programs such as today's symposium. Daniel's support and guidance through M-BoCA has helped numerous faculty achieve grants and expertise in the field of aging research, and M-BoCA would not be what it is today without Daniel's hard work and sacrifice. His memory is especially poignant today, as this will be his first year that he will not be a part of the symposium. In the midst of a bit of sadness and remembrance of Daniel's legacy, we also want to celebrate and excitedly anticipate the next chapter of M-BoCA under Daniel's successor, Dr. Latif. So thank you today for attending the Daniel Goldstein M-BoCA Symposium today and for reflecting and remembering our friend and colleague, Dr. Daniel Goldstein.
So in the spirit of collaboration and enhancing collaboration between the centers, our first speaker today is Dr. Victoria Blanc, who is the director of the Central BioRepository at the University of Michigan Medical School. She leads the facility, which plays a key role in supporting biomedical research by providing high-quality, well-annotated biospecimens for basic, clinical, and translational research. Under her leadership, the CBR has expanded to store more than 800,000 samples from various studies. The repository also provides critical resources for projects in areas like precision medicine, genomics, and molecular medicine. Before joining the University of Michigan, Dr. Blanc held leadership roles in the biorepository management at Astrand, where she helped the facility achieve national accreditation. Today, Dr. Blanc will be talking to us about the biospecimen resources in the Medical School Central BioRepository.
Good morning, everyone. Thank you so much for inviting me. I'll let Bruce do his wonderful job here. I'm very honored to be presenting today at the Daniel Goldstein lecture. Daniel and I worked together very briefly when he established a biorepository at the CBR, and it really was an honor to meet with him and work with him, and I very much enjoyed that time together. I also want to say thank you to Dr. Latif. Thank you also to Dr. Marian Hoffman for alerting me to this symposium, and of course Bruce for everything that he's done here today. Okay, so the CBR is dedicated to facilitating healthcare discovery, research discovery, by providing high quality, highly annotated biospecimens donated for basic clinical and translational research. We do this by partnering with PIs at the institution, their collaborators in consortium projects, departments, anyone who is interested in creating a shared resource for distribution is welcome to participate in the CBR so that we can honor and carry out this mission. We also provide services in support of this mission, such as DNA isolation and kit production for those types of collections as well. The guiding principles behind the CBR are to speed research by enhancing the availability and access to biospecimens. If the specimens already exist, you don't have to go out there and collect them on your own, so you should theoretically be able to get them much quicker. We also eliminate duplication and allow more efficient utilization of resources. The consolidation aspect really does make it easier for us to operate at scale. We do allow our researchers to focus on research, not biorepository management. None of our PIs were trained in how to manage a freezer, and nor do we want our graduate students fussing with the compressor on a freezer, so we'll do that for you. We do protect our valuable collections through properly managed infrastructure, so everything that we do is checked, managed, checked again, checked again. We are a very protective environment. And because the samples are available not only to the original researcher or the research team, we do allow enabling of an amplification of the research beyond the original question asked. All biorepository programs that are started at the University of Michigan through a University of Michigan PI or department are use the biorepository pamphlet that I have up here. So there's an informed consent document that is consolidated down to one page, front and back, and the affiliated pamphlet that's shown here. This enables our patients to understand quickly what they're getting involved in when they're invited to participate in a biorepository. The pamphlet and consent allow for broad use of biospecimens, future unspecified research. Genetic studies are included, and we do have an NIH certificate of confidentiality. That may not mean very much for those of us who are funded by NIH, because now NIH has wisely offered a global certificate of confidentiality for any studies that are funded through NIH, but those studies which are not are also covered through our own NIH COC. We also allow the sharing with anyone, and that is explicitly stated in the biorepository consent. And that includes with commercial vendors. So we have an information management system, does everything on the left-hand panel. We provide specimen provenance through every step along the way in the system, so anybody who touches the sample is recorded. All of that's done through logging in. There are permissions for certain elements within the system, so our research coordinators are accessing PHI as they enroll our patients, but that information is not shared with somebody who's not affiliated with the team, so there are role-based permissions for information. We do connect with MyChart, so MyChart can feed right into LabVantage in terms of the MRN, the participant name, and demographics. So that kind of information is pulled right into the system. One thing I wanted to highlight here is that we have a subject ID. This is one-to-one with the MRN. It's another code, but it does allow you to have continuity between our system and MyChart. As I mentioned also, there is the connection directly with the MRN. What you can see is that we have study A and study B. There are two participant IDs, but it is the same person. So within our system, somebody could be enrolled in a biorepository that's spearheaded by someone in the cardiovascular center, but they may also be in another biorepository program, and that is discoverable through the system. Okay. I have something on this screen that I have to get rid of. Okay. Next slide. So we also have physical infrastructure that supports all of our biorepository program. We have 2,800 square feet of storage, comprising currently 14 large cryogenic freezers. These are LN2 cooled freezers, so they are kept cold by virtue of physics rather than electricity. So that cryogenic freezers are cooled through a liquid nitrogen bulk tank that is outside the building, and the call for the LN2 happens automatically. We also have 18 minus 80 freezers, and we have storage at minus 20 at 4 degrees and ambient storage. We have space for an additional, or up to 27 minus 80 freezers, and up to 20 cryogenic freezers. We also have 2,200 square feet of laboratory processing facilities. This is where we do our DNA processing, for example. All of our aliquoting happens here. And we have staff who are trained in each of these techniques. Another service that we do often nowadays is PBMC isolation. All of our work is done under a quality management program, which offers significant benefits to the program. Our data are supported by evidence. Our data quality is supported by evidence and quality control metrics. We monitor our systems for when things or systems go awry. So if we see a blip, we're able to say, that doesn't look right. So we go in and we investigate what's happening. We provide confident stewardship of donated biological specimens. So if it happens, it's written down, it's recorded. We make sure that everything that we do is discoverable down the road if we need to. We use our systems to do CAPA management, that is corrective action and preventative action. So that helps us prevent mistakes down the road. So we're a continuous learning opportunity type of environment. Reduction of wasteful activities, as I mentioned, the consolidation lowers overall costs. And this recording of everything provides a living history of everything that's happened to the biospecimens, to the facility itself, and interactions with our investigators. This becomes way more important now that we only have 2 years to keep track of our email. So we have it for the entire lifetime of the biorepository. So how we help overall is we provide biobanking services, specimen access, and research enterprise services. So we provide safe storage, as I mentioned, connection to the electronic health records, and then the RDW and other databases that link to the biospecimens, like REDCap for our studies. We provide that standardized broad consent that I mentioned. We also provide an opportunity for governance and compliance. I'll get to that in a moment. And we provide a method for sample sharing, so the infrastructure to provide samples to others. Ready access to large numbers of samples allows us to have statistical power in our downstream research so that we can answer bigger questions. The connection to the electronic health record and these other databases provide deep clinical and phenotypic data that are accessible by the study teams, as well as longitudinal and outcomes data. Hopefully all of this provides the opportunity for faster publications, right? Again, if you have everything at the ready, you don't have to go out and get it. You just have to do the experiment. You can publish. In terms of the research enterprise, we provide economies of scale through the consolidation of the biorepository resources. Enhanced compliance, we do have some review processes for IRB approvals, compliance with state, federal rules and regulations. We also provide competitiveness for grants. I have been asked to write letters of support for grants. I'm happy to do that at any time. Also, the infrastructure of the CBR is described, and you can get access to that on our grant services and analysis website. We also provide synergies across our core infrastructure. We have outstanding relationships with our other core facility team members. So one example is the Advanced Genomics Core. We have a seamless workflow between our facility and theirs. So overall, we provide security, quality, access, experimental power, economy, right? Economy of scale, economy for our organization, picking up grants, increased direct costs, indirect costs, all of that. So, all right. I can do anything after this talk. So here's our portfolio at a glance. We have 44 biorepository studies that we are supporting, of which 4 are NIH consortium biorepositories. We have 2 external-only projects. They do have affiliation with the University of Michigan. There are PIs affiliated here, but they are part of a consortium, and so we are serving the consortium. That's CureGN and the Drug-Induced Liver Injury Network. These are both NIDDK. We do some internal service only projects. As I mentioned, we do DNA isolations for some of our faculty at scale. They don't have the automation to do the number of DNA samples that they're collecting, so we can do that. We also do nasal swab aliquoting in partnership with pathology, who is just across the hall from us. That obviously, well, maybe not obviously, but that did come about through the COVID pandemic. And we are continuing to do that work with COVID samples now and flu samples that come in. This is what our inventory looks like in a more detailed view. We can get even more granular and ever more granular on this, but on the right-hand side, you can see the primary research cohort. So this is our studies rolled up into a title characterization so that if I had all 44 on here, you would be blinded by the small print that I had to put up there. We have over 800,000 biospecimens from over 100,000 patients, participants. So we have more than one sample per patient or per participant. So that enables longitudinal access to the samples. On the left-hand side, you see the pie chart. The overwhelming number of samples are plasma, urine, serum, and DNA. And then everything else sort of falls into smaller percentages. But there are a lot of different types. They're down at the bottom. You can also see these sample types on our website as needed. I mentioned governance. So the CBR has an advisory committee. This is comprised of our faculty leadership, our deans, and experts in biorepository processes. We represent, or they represent, the stakeholders at large, so all of the University of Michigan Medical School. They help me and my team with policy and guidance setting. Some of these policies are on the side here. Technical direction, what do I do if I have this type of procedure that somebody is looking for and we've never done it? Do you know of any experts? They do an assessment of our program. So each year we provide an update on the metrics, the numbers that we have in the biorepository, production units. Our quality management program is reported up through our governance or advisory committee. I should mention that the CBR is accredited by the College of American Pathologists Biorepository Accreditation Program, and that program is deemed equivalent to a CLIA type service. So if someone is looking to do a CLIA test on a sample that is supplied by the biorepository, they are able to do so because of our deemed status. Each individual biorepository program is required to have an oversight committee per University of Michigan Medical School policies. So that oversight committee provides scientific and clinical content experts for the biorepository itself, that program. They provide access, utilization, and distribution decision-making. So they overlook or oversee questions of feasibility, scientific merit, statistical power, and appropriate usage of the biospecimen. So anytime you are seeking to access biospecimens through the biorepository, we will facilitate conversations with that study team for their oversight so that they can make a decision about the sample usage. They also serve themselves with their own KPIs and metrics. Okay, I won't go into too much detail on those. Just know that those exist, and anytime you're accessing biorepository samples, somebody's going to ask you about these things. A biorepository near and dear to our heart, and near and dear to yours, I assume, is the Cardiovascular Improvement Project. Dr. Marion Hoffman-Bowman is the PI. This study is aimed towards elucidation of the genetic and environmental causes of cardiovascular disease. Some of those diseases are listed below. There's over 7,900 participants in this biobank. It's been running for quite a long time. So again, there's lots of longitudinal data affiliated with these samples. You can see the sample type on the and the number on the right-hand panel, 11,000 samples across 7,900 participants. The CHIP biorepository has its own webpage here, and you can see it down here. And I know that there was a poster up earlier for this symposium that you can look at. Another cardiovascular-based biorepository at the CBR is the Vascular Disease Registry for Studies of Dysplasia-Associated Arterial Disease, or DAD. Santhi Ganesh is the PI there. She is looking to identify the genes and cellular molecular pathways involved in arterial dysplasia. She has over 11,000 cases. She has also 1,300 control patients who are part of the biorepository. There's 1,678 participants. The DAD study is a Taubman study, so that is funded by the Taubman Institute. Another cardiovascular-related biorepository is the Immune Precision and Solid Organ Transplant called IMPRESS. This was started by Daniel Goldstein years ago. He wanted to look at the effective immune suppression strategies for solid organ transplant recipients to minimize rejection and post-transplant infections. This study includes patients who are receiving heart, liver, kidney, and lung transplants. Since Daniel has turned over his responsibilities to Dr. Abhijit Naik a few years ago, it is primarily kidney disease or kidney transplants now that are being recruited into this program. But there are some heart, liver, and lung transplants also. This has tissue affiliated, so there's biopsies taken at multiple time points. There's 457 participants right now with 28,000 biospecimens. This program also works with the CyTOF core, so there's CyTOF data affiliated with a subset of these participants. And one study that is not necessarily a cardiovascular study, but I do want to highlight to you is the Michigan Genomics Initiative. This is a precision health program. It's a collaborative effort among physicians and researchers and patients at U of M, where we are combining the electronic health record with GWAS data, genotype data. So each of these participants has provided a DNA sample, which is why I did not put the biospecimens on the right-hand side, because all of the biospecimens are DNA. Instead, I put the phenotypes that are represented in the cohort. The genotype data is delivered using the core exome genotyping array and or the Infinium global screening array. These data are available to our investigators at the University of Michigan. You put in a request through the Precision Health webpage. Additional genotypes on all of these patients are imputed on the Transomics for Precision Medicine, or the TopMed reference panel. Santhi Ganesh is one of the big users here. She has done a fair amount of work using Michigan Genomics Initiative data. Before she left, Kristen Willer also did a fair amount of work with the MGI, and I know that Chip has a fair amount of genetic data. So combinatorial access to these resources should produce some nice outcome data or research data. In general, if you're looking to access biospecimens from the biorepository, you can do so using Data Direct. That is a shared resource sponsored by the Office of Research at Michigan Medicine. This is a self-service tool. You need Michigan Medicine access, so formerly level 2 credentials, but now you have to be affiliated with Michigan Medicine to access PHI or PII. You need peers training, and you need a Hume number if you want to see information at an individual level. If you are just looking for aggregate data, you can access the information without a Hume number specifically. So we interact with Data Direct, the research data warehouse. Our biospecimens are discoverable in aggregate through Data Direct. Once you have identified the cohort that you're interested in, we can help you identify the specific samples. You would write a use proposal with your objectives, hypotheses, methods, et cetera. And we would share that use proposal with the study team who has oversight control over the biorepository. If you're looking to access MGI data itself, I have provided the email there for you. Okay, I want to say thank you to my team who are outstanding. None of this work happens without them. They're really a bang-up group of people who do a wonderful job. Our partners at HITS really help us manage our information management system. We couldn't do it without them. Steve Kunkel, Carl Jepson, and Cassie Wong obviously are our sponsors. And we do a lot of work with facilities, so they need to be mentioned here too. So thank you for your time. I think I have one question, so if there's any questions, I'll take that.
So Victoria, first of all, you are an institutional treasure, and your biorepository is an institutional treasure. So it's just, grateful for all you do. It's very valuable, probably to industry as well, because we have biorepository and EHR data connected. How does industry approach Michigan, and how do we allow it? Obviously, it's super useful for those of us who are researchers, but what about external partners?
Yeah, that's an outstanding question. So an industry partner must have a faculty member at the U of M who they are working with in order to gain access to the resources in the biorepository. And then there is a biospecimen and data, or a data and biospecimen use committee through which all requests for industry access must go. There's a review of the project, the types of information that are being requested by the industry partner. If there are certain parameters met, there's sort of a, yes, this meets all the checklist requirements, and it can go through after sort of basic approval. If there are more complicated requirements, then the full committee will discuss the application by the industry partner for access. So yes, there's a process, Data and Biospecimen Use Committee, and that is comprised of folks who are knowledgeable, like in industry partnerships touching Ketterpal. Steve Kunkel is, of course, on the committee, data. Aaron Kaliba, who runs the data office, myself, others who have experience working with industry. Yes.
Hi, Victoria. Thank you very much for your presentation. I would like to know if some of these cryopreserved PBMCs, they're actually viable for reprogramming into iPS cells, because we're frequently asked about the size of our biobank at the CVC cell regeneration core, and although we carry some disease-specific cell lines, we can't cover everything that is available out there, and then the CBR would be like a wonderful resource to obtain viable cells that can be reprogrammed to each, to the interests of each specific researcher.
Yeah, so I don't know the answer to that, unfortunately, because I don't do science anymore. But I can tell you the method through which the cryopreserved PBMCs are preserved, and if that helps. We have 3 different methods, so I won't go into them here. But if that helps you assess whether or not they are iPSC viable, or whatever you said they were. I don't know. I'm sorry. That should help you, I hope. So I don't have a good answer for you at this time.
One last question, and then we'll...
Did I see a question?
Yeah,
Yes, oh.
Victoria, I'm going to follow that up. So the cryopreserved PBMCs in the central biorepository is quite low compared to the total number of samples. But then in Daniel's specific example, the cryopreserved PBMCs was really, really high.
Yes.
So I'm assuming that's because either someone provided staff or storage to make that happen. And what's the rate limiting step for expanding cryopreserved PBMCs?
So Daniel set up the biorepository with the intent of isolating PBMCs. We started doing the PBMC isolation processes with Daniel. He had somebody else do it, and then we brought them into the CBR. And since then, it has become a more requested service. And so the number of studies who are using PBMCs and isolating PBMCs for their research has gone up in recent years. So I think we had none when we started, and now I think we have 5 or 6 studies that are doing it. So that's how it's getting populated.
And so are those being stored centrally then in the CBR now?
Yes.
Yeah.
And those are all at LN2 vapor phase.
Thank you.
Okay. Thank you so much.